Good evening to all. On behalf of Veridian Microlab I Private Limited, I welcome everyone for this wonderful webinar evening. Thank you for joining with us for today's webinar. I hope everyone will enjoy the CRISPR-Cas9 technology. It is one of the unique and cutting-edge research technology which is evolving around the world now. It is having the ability to precise, edit, and change any part of an organism genome. So let us start the basic session and in the CRISPR technology and the basics and how it is discovered, the timeline and history, and how bacteria is precisely using this wonderful technique to making it as a immune response. Followed by we have a session of ethical issues and social and legal issues when we are using a CRISPR technology. Other than that, we are also going to learn about the applications of CRISPR technology. Let us move on to our presentation today. CRISPR technology. So CRISPR technology is having a precise on-target activity as well as specificity to make a cut on target gene. So whenever we need to knock out or knock in or creating any transgenic animal model or a plant model, so we have to precisely make a cut in the particular gene target so that in that place we can able to do our edit to create our desirable modified organism. So to precise edit and change the particular part of an organism genome, this CRISPR technology will be very much useful. So it is having a wonderful advantage like on target activity. It can have the ability to make a precise cut. Followed by it is having a specificity where it has to make a cut and where it does not have to make a cut. So it is having a distinguishable characteristics and this characteristics making it more precise to use in worldwide genome editing. So let us see the basics of CRISPR technology. So CRISPR, the word CRISPR stands for clustered regularly interspaced short palindromic repeats found in the genome of prokaryotic cells. So it is found both in bacteria and archaea as well. Initially, it is discovered in bacteria and archaea. So these are all nothing but the repeated units. As we know already, in eukaryotic cells, we have repeats. So that repeats generally we call it as tandem repeats. It might be varied from a length. If it is a short repeats, we call it as a short tandem repeats. If it is a long one, we call it as a long tandem repeats. So based on the length, we are distinguishing. And according to that samely, in bacteria, they are also having a spaces. So that spaces regions and the palindromic repeats, we called it as a CRISPR. So this palindromic repeats, what is mean by palindromic? As we know, in restriction endonuclease mechanism, this palindromic repeats are nothing but the sequences which can able to will be the same even though we are reading it from left to right or right to left. It is nothing but in molecular biology we can term as 5' to 3' direction or 3' to 5' direction. So in either way, if we read, that particular sequences will be looking like the similar one. So that is one of the unique characteristics. And this CRISPR-Cas9, actually this CRISPR is a combination of complex molecules. So those complex molecules are nothing but the spacer region and the Cas9 protein, that is CRISPR associated protein, and another one is the guide RNA. These three are aided together or they are worked together to make a precise cut in a genome. This CRISPR-Cas9 are generally or in naturally they are acting as a adaptive immune responding system in prokaryotic cells. For example, if there is any invading of a virus or a bacteriophages or any transposable elements into the bacterial cell, now this bacterial cells will detect the infection. Once there is a attachment of that particular elements into that bacterial cell. automatically it recognizes something is infecting. So at that time, it will make and it will express the CRISPR array. So that CRISPR array will detect and they incorporate that particular gene segment of that viral particle, and they incorporate that uh, short stretch of a gene into that bacterial genome. Once they incorporating, that incorporating will express a protein, so that we called it as a CRISPR RNA, and followed by there is the transactivating RNA. Once there is a combination of these two, and these two combine together to form a guide RNA. Now this guide RNA will recognize the PAM sequences on the CRISPR array, followed by the recruitment of the Cas9 will make a precise cut. This is actually the outline of the CRISPR technology. So this is how the bacterial cells protect themselves against that foreign particles like viruses and bacteriophages. In case if that particular bacteria, that is the bacteriophages, which is already infected a prokaryotic cells. So if that bacteriophage again they are infecting that bacterial cells, automatically the CRISPR array will get expressed and it starts to make a precise cut. Once there is a fragmentation of that nucleic acid of that particular invaded viruses, it will leads into a death of that viruses or inactivation of that viruses. So thereby they can't able to further infect the bacterial cells. So this is how they are involved in adaptive immune response. Now, this natural responding is exploiting by humans to make genome editing. Now, let us see the basics and what are all the complexes involved in CRISPR technology one by one. So, first we'll see the history. Actually, there is a timeline for the discovery of that CRISPR technology. So in 1987, the first discovery of CRISPR is identified in E. coli by the scientist Yoshimi Ishino and his term. So, who accidentally cloned an unusual series of repeated sequences, that is the interspersed with the spacer sequences, while doing a gene analysis, which is responsible for the conversion of alkaline phosphatase enzyme. So during this event, they are accidentally identified the CRISPR. Some short stretch of repeated sequence they have identified. Followed by in 1933, researchers or a research work led by G.D. van Emden in Netherlands, they discovered different strains of Mycobacterium tuberculosis. So in that work, in that working place, they had different spaces sequences, or they have identified different spaces sequences between DNA repeats. So they have a wonder, and they are characterized that particular tuberculosis strain based on that identified spacer region. Now this technique is widely we called it as a spoliotyping, and subsequently these sequences were identified not only in Mycobacterium tuberculosis, many other bacterial species is also having the similar repeated sequences. Until that, they have not identified actually that is a CRISPR. They have identified some repeated sequencing is present. So up to that only they have discovered. In 2000, the scientists Mojica and the co-workers, actually they have discovered this particular repeated units are involved in the function of bacterial adaptive immune response when there is a infection of foreign particles. So actually they have identified that is having a role in immune system. So they discovered that viruses cannot infect bacteria homologous spacer sequences unless they suggesting that these sequences will have a play role in adaptive immune system of prokaryotic cells. In 2012 and 2015, so once there is a complete identification and a frame out of what exactly the CRISPR, so the researchers and scientists are now turned into exploitation of that particular CRISPR-Cas9 as a genome editing tool. So in current world, still now we are spotlighting. It is one of the challenging genome editing tool in all biological science field. So it is applicable in therapeutic applications like cancers or blood cell editing tool. And other than that, we can also using this CRISPR in agriculture to make a transgenic crops which will give eye yield or superior traits or superior quality of that particular fruit or seed. So, based on that particular gene, what we are targeting, it might be have some economical use or it is having some therapeutic application. We can make a edit or mutate that particular gene target to create our desirable genome editing. This is the overall timeline of the CRISPR technology. See, from the identification, the discovery of this wonderful tool takes around a time period of 1987 to till now. So just think how unique it is. So let us move to that um, actual mechanism of CRISPR in bacteria. Actually, as I said, this CRISPR-Cas9 technology is one of the important pathway which is acquired by the bacterial cells to make an adaptive immune response when there is an infection of a foreign particle. So CRISPR-Cas9 was previously discovered in bacteria and archaea, as I said, as an adaptive immune response system. So this CRISPR loci is observed that 40% presence in bacterial sequence and 90% abundance in archaea sequence. Possibly, if you see or if you did any Sanger sequencing of this particular CRISPR array in bacterial cells, you can able to differentiate this CRISPR array is not only present in one locus, it is also presented more than one locus. So that is also they have reported. So this particular CRISPR array having the mechanism when there is an invading foreign genetic materials, that is nothing but the bacteriophages or virus or transposable elements or any undesirable nucleic acids. So generally, those having a infective nature. So that are cleaved with the help of the endonucleases called case nuclease. Actually, this case is acronym for case associated protein. It belongs to a category of nucleases enzyme. We know nucleases are categorized into two categories. One is endonuclease, another one is exonuclease. So the endonuclease having the mechanism to make a cleavage in between the double strand. When in the case of exonuclease, it can able to trim or make a cut at the terminal part of the double-stranded nucleic acid. So that makes a sense and differences between endonuclease as well as exonuclease. Now come to CRISPR. This case nucleases belong to endonuclease category. Once this expression of endonuclease or the recruitment of endonuclease Cas9, it automatically captured that particular genetic material or a short stretch of DNA fragment from the invader, and it is get integrated and incorporated into the CRISPR locus of the bacterial cells in the form of spacer sequence. Actually, the spacer sequences are nothing but the it's similar to that introns, how they are like a spaces in between exons. As like in CRISPR array, these are all the sequence. In that particular spacer sequence, the captured genetic material of that invader is getting incorporated in between the conserved repeated sequences. So these conserved repeated sequences will not undergo any mutation according to evolution. It will stay as it is. In the spacer sequences only, there will be a presence or adaptive of mutations in relation with the evolution. So the integrated spacers act as a template to form or synthesize a CRISPR RNA. Once there is a formation of a CRISPR RNA, that will form a complex of trans-activating RNA. Now, this CRISPR RNA and trans-activating RNA combine together to form a complex. Now this complex will aid or nothing but the having a role in guide RNA. Actually, the guide RNA is nothing but it is a combination of CRISPR RNA, that is pre-processed and matured CRISPR RNA and trans-activating RNA. Now, this guide RNA will search the DNA strand according to the presence of a PAM sequence. So this PAM sequence, we call it as a protospacer adjacent motif. In upcoming presentation, we'll see. So this PAM sequences is recognized by that guide RNA. Once they are getting recognized, it attached to that particular template strand and it further recruits case nuclease enzyme towards that. And once there is a complex formation of CRISPR RNA, transactivating RNA and guide RNA, now this case 9 protein will have the activity to make a cleavage of double strand. That is, the double strand breaks is possible. Once they bound, as I said, the case 9 cleaves based on that complementarity of the sequences which is present on the template strand, and through its with the help of two domain. Actually, this case protein is having two domain, HNH and RUBC1 like nuclease domains. With the presence of these two domains in the case 9 molecular structure, they can able to make a precise cleavage in the bacteria in the foreign particle genetic material. So this is how the mechanism or the path, the steps of adaptive immune response by the CRISPR, which is utilized by the bacterial cells. Actually, if you see here, as I said, this is the overall CRISPR locus. So this CRISPR locus, if you see, we have a array of clustered palindromic repeats here, clustered regularly interspaced palindromic repeats array. You see here, these are all the spacer regions. So in this spacer regions only, the viral, that is the bacteriophage genetic material, that short stretch of genetic material getting incorporated in this region. Once there is an incorporation, this region will get expressed on, and it is followed by the gene expression of CRISPR RNA and transactivating RNA. Once there is the gene expression, there is the synthesis of these two proteins and they will form a complex. Finally, it releases a guide RNA. Once there is a guide RNA synthesized, it starts to read the strand according to complementarity. Once they found that complementarity, now this case 9 protein will recruit there and it make a double strand cleavage. So once there is the cleavage of the viral genetic material, it is not able, or they can't able to longer to infect that particular bacterial cells. Automatically they degenerate inside the bacterial cells. So now, how this CRISPR Cas9 hijacked for genome editing? Why? Because it is having a user friendly, and it is and its stringency, nothing but the consistency. We can able to design according to our own desirable creation of the transgene. That is one of the unique characteristics of CRISPR. Another one is rapid metabolic engineering. In short stretch of time, we can able to create a much more combination of a guide RNA. So actually, this guide RNA is not is the important one which will guide that case 9 protein to make a idea where to cut, where not to cut. So this guide RNA is rapidly engineered with help of lots of tools are available, like like editing tools are available. So according to that, we can able to synthesize and design our own guide RNA. Another one, as we know, until today, we have n number of bacterial species have been identified and they are sequenced and they have submitted in NCBI till now. There are also not unidentified novel species are also existed in various living habitats. So, in response to that identification of that particular bacteria, we can even more can able to identify very novel CRISPR array and CRISPR Cas9 mechanisms of bacterial cells. For example, if you take the CRISPR Cas9 system of E. coli, it is different. And if you are taking an example of Streptococcus pyogenes, so that CRISPR Cas9 mechanism is entirely different. So the mechanism and the presence of and category of Cas9 proteins. So that makes a sense and differences between the CRISPR Cas9 system in one species to another species of the bacterial cells. So based on the identification of a novel bacterial cells, even though we can able to identify discovery of novel Cas9 like CRISPR systems from wide diverse of microbial environments. If you take a CRISPR technology, we have two systems. One is model host organism, another one is non-model organism, which is nothing but model host, that is the E. coli. This is one of the universal gene expression or expression vector. Actually, we say in molecular biology, E. coli is one of the universal expression vector for gene cloning, gene editing, for bacterial chromosome walking. So for most of the experiments, we are using E. coli as an expression vector. Other than that, in rare cases, we can also use, particularly I'm saying in CRISPR, we can also use this non-model organisms like Clostridium, Bacilli, Streptococcus, Cyanobacteria, and Streptomyces. So those are also we can use. And let me see disadvantages of CRISPR Cas9. Even though it is having a wide range of advantages, all not only CRISPR, all technology, it should have some own characteristics, disadvantages as well as advantages. So based on the disadvantages over disadvantages, we are choosing that particular technology for the editing and other applications. The disadvantages of this CRISPR is costliest one. Still now, the designing and synthesizing of that particular guide RNA is one of not cost affordable. Followed by it is another disadvantages is off-target cleavage. Actually, what is mean by off-target cleavage is nothing but when there is a Cas9 protein, if that particular CRISPR array, actually in the sense that if the CRISPR array is not work ideal for all genome editing. So some particular genome editing, it is applicable. In some cases, it is not advisable to use that same CRISPR array for all genome editing. So in that particular cases, the not suited Cas9 nuclease will make a unprecise cut in the non-targeted gene region. Actually, we are using this CRISPR Cas9 to make a precise cut in a target region. If that particular CRISPR array is not working in some living organisms, automatically that will lead to the cleavage of untargeted region. Just imagine if that untargeted region is spacer, it is very luckily we don't have any phenotypic resemblance of phenotypic errors. But in case that particular cleavage is happened in the necessary gene target, for example, if that particular gene is synthesizing a growth protein, or that particular protein is involved in cellular respiratory of the prokaryotic cells, or the organism which we are editing. So in that case, it will create a serious errors in that phenotype as well as the genotype of an organism. So we have to very precise while choosing that CRISPR Cas9 system. And another one, it is a time consuming. Actually, for designing a guide RNA, at least we have to make some combination, random combination. Until we are moved to random combination, we can't able to identify which particular guide RNA is precise and ideal to our editing. So that we have to make a design and we have to make a synthesize. For that it will take some around month, 1 month to 3 months, based on that person who is already already get experienced or fresher one. So based on that time consuming method will get differ. And limiting the widespread use, and we can't able to produce this CRISPR Cas9 in a large scale manner. So still now we are in the place to utilizing that CRISPR Cas9 for our R&D or any clinical trial works. So and also it is considering as a high throughput study. When we are doing some high throughput studies, it is creating, if off target cleavages or any unprecise get, automatically it will destroy that particular high throughput studies and cutting edge research practices. So these are all the disadvantages of that CRISPR Cas9. But over on that, still now it is having a much more advantages than disadvantages. So that we are using this CRISPR Cas9 for wide range of genome editing. So let me see the components of a CRISPR Cas9. So first component of the CRISPR Cas9 is the Cas9 protein. So Cas9 protein is stands for Cas, that is CRISPR associated protein. So this CRISPR associated protein is having a molecular weight of 160 kilodalton, and it is generally called as RNA guided endonuclease. Why? Because it is followed that guide RNA to give an idea where to make a cut. So that makes a sense, and it is having a name of RNA guided endonuclease. So that can be used for site specific cleavage or site recognition cleavage of invaded viral double stranded genetic material. It might be either double stranded or a double stranded DNA or a single stranded RNA. It not make a sense that actually it recognizes that particular genetic material. Once it recognizes, so there is a complex formation of that CRISPR RNA and transactivating RNA. This Cas9 RNA will make a cleavage there. So that is the crucial role of the Cas9 protein. As it belongs to a endonuclease family, it is having a two major lobes, or we called it as a domains or a functional domains. So that functional domains, one is alpha helical recognition lobe, that is the REC protein. The acronym of REC is response regulator with the key Y-like receiver domain, and nucleus lobe is nothing but the nucleus domain. It belongs to a endonuclease domain, and it is having a name based on that activity. So the REC is comprised of 3 alpha helical domains, and these 3 alpha helical domain sequences is not having any similar sequences with the already existed or identified protein in prokaryotic cells. This NUC, that is nucleus lobe domains, other than this domain, that it also includes as other 3 domains: RUBC domain, HNH domain, and C terminal domain. This NHN domain is nothing but it represents the composition of the amino acids histidine and arginine. So the presence of this amino acids, it is having the name of HNH. And C terminal domain. In this C terminal domain only, there is a short stretch around some 2 to 6 base pairs of PAM sequences will be present. So the acronym of PAM is nothing but protospacer adjacent motif. Based on the presence of that PAM sequences only, this guide RNA will bind and they will form a complex. Once there is a formation of a complex, it will further recruit that Cas9. So further steps will be getting activated. The 2 lobes are connected by means of the presence of 2 linker sequences. So that linker sequences will make a interface between the 2 lobes. So that 2 sequences are arginine rich bridge helix and disordered linker sequences. These 2 are the bridge linker sequences will form the bridge between these two domains, rec and nuc. If you see the diagram, you can able to identify the overall case 9 complex um, activation and structure. So the single guide RNA bound with that DNA, and thereby it is forming a single guide RNA DNA complex. Once it bound at the interface between these two lobes, the C-terminal domain contain that PAM interaction residues, which is necessary for the recognition of the guide RNA. So that is identified by guide RNA, and it recruit that case 9. The resulting reaction is the nuclease activity. Thereby, it will form a double strand breaks in the DNA of that invader genetic material. Another component of CRISPR Cas9 is CRISPR RNA, trans-activating RNA, and a guide RNA. So, in bacteria and archaea, the CRISPR RNA and trans-activating RNA form a complex which act as the homing device. So, now this homing device direct or guiding that Cas9 nuclease to make a cut in the invader nucleic acid. By combining these trace RNA by combining this trace RNA and CRISPR RNA into a single synthetic guide RNA, resulting in acting as a one component system into a single synthetic RNA, resulting as one component system. It not only simplifies the whole experimental design, it also yield a equal and a great guiding efficiency to make a precise cut in a genetic material of an invading bacteriophages or a viral particle. As I said before, the combination of this trans-activating RNA and a CRISPR RNA altogether will form a complex that resembles the guide RNA. So there is a different, as per our need, we can able to construct a wide range of guide RNA based on our experimental design. Once we are designing, we are moved to that testing side. So in testing side, we can test which particular guide RNA is optimized and ideal to our genome editing experiment. The editing rate will get increased in related with precise designing of that particular guiding RNA. So how much we are reducing our errors while designing guide RNA, that much we have a efficiency increases in the editing rate of our genome. The another component of CRISPR technology is the PAM recognition site. Actually, this is one of the important recognition site. Without this, guide RNA and further cleavage reaction is not at all possible. So, what is mean by PAM? PAM is nothing but protospacer adjacent motif. Case 9 recognizes the PAM in the CRISPR repeat sequences mainly to recognize self and non-self genetic material. For example, if there is no PAM sequences, automatically if guide RNA it bind with the template strand of the DNA, it doesn't have the idea where it is binding, either with bacterial DNA or a viral DNA. Actually, the immune response of that CRISPR is based on to make a cut in the viral DNA, not in a bacterial DNA. If it is making a cut in a bacterial DNA, both the viral particle as well as the bacterial cell will lead to a death. To avoid that, that CRISPR array is having a short stretch of sequences, that is the 2 to 6 nucleotide length, that we called it as a PAM, that is protospacer adjacent protein. Naturally, These PAM sequences are located in the downstream of the target region, that is the 5 prime. So immediately it followed the 3 prime. So immediately it followed the target region sequences. For example, if there is a 100 nucleotide stretch of it, your target region where you want to make a cut. So this PAM region should be present immediately followed that target region. PAM recognition sequences, once it's there, it activates nucleus domain of case 9. As I said before, rec domain and NUC domain, it activates that two domain to make a distinguishing between self and non-self genetic material. It prevents the CRISPR loci from being targeted the self genetic material. In order to avoid that, the CRISPR array is having this PAM sequences. So PAM recognition sequence will get differ from one species to another species of the bacteria. As I said before, CRISPR-Cas9 system is having a wide variety of and differences in different bacterial cells. E. coli CRISPR array is not same to that of Streptomyces species CRISPR array. So there is always a differences, not that much difference, but still there is a differences like 1% or 2% differences between the repeated units of that CRISPR array. And the most commonly used CRISPR array system still now for genome editing is the type 2 category. Type 2 category. Actually, CRISPR-Cas9 is categorized into 3. Category 1, Category 2 and Category 3. Now we are using CRISPR-Cas9 technology belongs to a Category 2 type, which is isolated from a species name Streptomyces pyogenes. So this particular nuclease recognizes a PAM sequences of 5 prime NGG on the immediate 3 end of, that is the 3 prime end of the guide RNA sequences. So this guide RNA sequence will recognize this PAM sequences and it will make a bound with the template strand. So this PAM sequences will get differ from one to one species. So this is the PAM recognition site sequences of different species. If you take a look in Staphylococcus aureus, it belongs to a subtype of 2 and it is having a PAM sequences similar to pyogenes. And Pseudomonas aeruginosa, if you see here, it is having PAM sequences recognition of 5 prime CC. And E. coli, if you take a look here, it is having a recognition sequence of AWG. These are all nothing but the coding of nucleotide sequences. A will code for adenine, U will code for uracil. R, in case if you find a R, it is nothing but it might be either a purine, adenine or a guanine. If you find Y, it belongs to a pyrimidine class. It might be either a cytosine or a thymidine category. If you found N, it is nothing but any nucleotide. So there must be a combination. If you see here NGG, it is not particularly, it should have a adenine only. But these guanine and guanine will be started. And instead of this N, it is representing any nucleotide. So in that place, adenine will be there, or guanine will be there, or cytosine will be there. Based on the species, that region will be changed. If you see here, this is the overall PAM recognition site sequences. If you see, followed by the target sequences, immediately the PAM sequences will be present. Once there is a recognition of the PAM sequences by the guide RNA, it automatically make a precise cut that results in the double strand breakage or in the genetic material. So let us see the mechanism of that CRISPR-Cas9 array in bacterial cells. Just take a look here. It is having a sequential steps. First is DNA invention of that particular prokaryotic cells. Second is embedding of the DNA that is incorporated into the CRISPR array by the prokaryotic cell. Once there is a achievement of a CRISPR, that is the incorporation, there must be a initiation of pre-CRISPR RNA transcription, followed by guide RNA synthesis, Cas9 complex activation, target that must be either DNA or RNA, and followed by that is a target cleavage. Final one is the inactivation of that invader genetic material. So this is the overall pathway of the CRISPR technology when it invades a particular prokaryotic cells. So in first step, DNA invention, when there is a foreign DNA from a virus or a plasmid that invades a prokaryotic cells, the second step will be sequentially getting on. DNA fragments from the invaded DNA particle of that particular invading elements getting incorporated into the CRISPR locus as a spacer. Once it is getting incorporated, the exact mechanism of incorporation is still remain unknown only. But once there is an incorporation of that CRISPR locus in a spacer, the cell consecutively get transcribes a repeat or a spacer group in the form of a premature CRISPR RNA. If you see here, there is a combination of a black box and a gray box and a red box. Actually, that red box resembles the gene which is getting incorporated from the invading particle of that viruses or a bacteriophage. And the black box represents the repeated units and the gray box repeated the spacer regions. Once there is a incorporation of this one and the transcription of that pre-CRISPR RNA, now the cell further consecutively expressed the trans-activating RNA, which is present within the CRISPR repeat sequences on the pre-CRISPR RNA. Now, RNase 3, CSN1, and other unidentified CRISPR-associated proteins will undergo modification of that pre-CRISPR RNA, that is for the formation of the maturation to form a complex or a duplex of CRISPR RNA, duplex trans-activating RNA, thereby it results in the formation of a guide RNA complex. Once there is a formation of a guide RNA, now the Cas9 will get activated. Until the binding of guide RNA, the Cas9 protein will be inactivated in condition. Once this guide RNA recognizes the trans sequences on the template strand and it recruits a Cas9, now this guide RNA and a Cas9 protein will get a complex formation. Once there is a complex formation, then the Cas9 protein will get activated. Once there is an activation of a Cas9 protein and complex formation of a guide RNA, now the target cleavage mechanism will initiated. Now the activated guide RNA Cas9 complex bind with that target template strand. Now the localization occur very rapidly and now the target cleavage is achieved. Now the CRISPR Cas9 protein cleave with the invading DNA and inactivate the viral particle from being further infecting the bacterial cells. This is how the bacterial cell switch on its adaptive immune system in response to protect itself against the infection of bacteriophages or any viral transposable elements. Now, once again, the same invading DNA is detected by the cell. The spacer DNA will automatically getting on and it express in the form of CRISPR RNA. Now this CRISPR RNA again will form a complex with the tracer transactivating RNA and thereby it form a guide RNA. Once there is a complex formation, as I said before, it recruits a Cas9 endonucleases to the template strand and it make a endonuclease cleavage activity in the double-stranded template strand. I hope everyone will understand the basics of CRISPR technology, which is widely used by the bacterial cells. So once now after the cleavage, what will happen? So once there is a cleavage, this endonuclease will create a two sort of ends. One must be a blunt end, another one will be a sticky end. So based on the category of endonuclease enzyme, the two formation will be possible. One is blunt end, another one is a sticky end. So when there is a cleavage, there must be a overhang. It must be from 5 prime to 3 prime overhang or 3 prime to 5 prime overhang. So that overhang should be ligated together by means of following a DNA repairing mechanism. This DNA repairing mechanism might be either non-homologous end joining method or homology directed repairing method. Actually, there is a differences between these two methods. The non-homologous end joining method breaks or ligated cleavage double-stranded breaks without the requirement of a homologous template. Whereas in the case of homology directed repair mechanism, it requires a template strand to guide a repairing mechanism. Actually, there is a many number of enzymes and proteins are taking a place to making a DNA repairing mechanism. Examples like DNA gyrase and like a REC protein, and these are all examples of that DNA repairing mechanism involved the protein. When in the come to the case of CRISPR Cas9, homology directed repairing method is having more advantage over non-homologous edge joining method. It is having this HDR method is having some precise genome editing advantages as well as the time consuming one also. And this non-homology edge joining is a very efficient repair mechanism. It is can able to see most active in all the cells. So it is also susceptible to make a frequent mutation error done by the insertion or deletions followed by any physical mutation or a chemical mutation or any DNA repairing errors while cell synthesis. So HDR is considered the dominant mechanism for precise double-stranded repair, but suffers from low efficiency because it requires very high number of sequence similarity. So for this reason, this homology directed method is widely accepted over non-homology end joining method, while in the case of CRISPR Cas9 technology. And if you see the steps of non-homology end joining process, it is having a three sequential steps. One is DNA end binding and bridging with the help of DNA dependent protein kinase. Second step, it is having a terminal end processing. It will process the terminals, that is the overhangs, to get ligated properly. Final one is the ligation. Once there is a precise arrangement and alignment of that DNA in response with the complementarity, the third step will be the ligation by forming a phosphodiester bond in the backbone of double-stranded DNA. It ligated the cleaved end and it make a precise ligation by with the help of a DNA ligase for ligation complex. These are all the sequential steps in the non-homology end joining method. If you see in homology directed method, once there is a formation of a double strand cleavage, it recruits some proteins, that is CHP and BRCA1 and MRN. So it further recruits that and they are involved in a resection process. Once there is a completion of the resection process, there must be a formation of a branch migration as well as the creation of holiday junction, that is the meeting point. Now this DNA polymerase will read the template strand and it add the complementary nucleotide to make a precise alignment of that double stranded cleavage, and thereby it repairing the breakage done by the nuclease activity. So CRISPR, after the cutting of that double-stranded DNA, the cell will acquire any method. Either it must be a non-homology end joining method or homology directed repair method. So based on the cell system and what type of Cas9 system we are using, both the repair pathway is acceptable one. Now we'll see what are all the errors is possible while we are doing CRISPR-Cas9 techniques. So for the most popular one is off-target cleavage. As I said before, off-target cleavage is nothing but it make a cut in unrelevant region or non-targeted region rather than a targeted region. To avoid that, researchers will use a wide range of mutation in a Cas9 protein, thereby they are creating an optimized one. That optimized one will minimizes that off-target cleavage errors. Let us see the example of this off-target minimized method. The example one is the Cas9 nickase. So this Cas9 nickase is nothing but is the same enzyme which belongs to the Cas9 nuclease enzyme, where the mutation is done in the region of DD10A and H840A mutation. Actually, this AA are representing the amino acids. So in domain of RUBCC1 and HNH like nuclease domain. As I said, these two domains are nothing but the nuclease domains, which is requirable and very important for the Cas9 to make a nuclease activity. So researchers are made a mutation in these two domains to get a mutant form rather than a wild type. Now this mutant form will form a cleavage in single strand only, not a double strand. So in this case, this CRISPR, when we are using a CRISPR-Cas9 system with the utilization of this Cas9 nickase instead of using a wild type, we must require 2 guide RNA instead of only 1 guide RNA. Why? Because this Cas9 nickase will make a cut in only 1 strand, not in a 2 strand. For this purpose, we require 2 guide RNA, and these 2 guide RNA should be in a much more precise and highly probable in sequence similarity with each other to avoid untargeted cleavage. So that in such a case, this Cas9 nickase will be utilized and it will make a single-stranded nick instead of creating a double-stranded breakage at the target site. Now this nick is normally normally repaired by either method. Either it is a non-enjoining homologous method or a homology-directed method. So, but usually this single-stranded is easy to repair rather than when compared with double-stranded breakage in the genetic material. It is quite easy for the cell to repair it. In this order, by the utilization of this Cas9 nickase, this off-target cleavage is minimized. This error is getting minimized. Now, let me see the application of CRISPR and the use of CRISPR over other protein-based genome editing tools. Other than this CRISPR genome editing tool, we are also having other protein-based editing tools like zinc finger nuclease, meganuclease, TALEN. TALEN is nothing but TAL and endonuclease, the combination. TAL plus endonuclease, so that we called it as a TALEN. So based on that unique characteristics of their own, as well as the advantages and disadvantages, this CRISPR is having more usable and advantage over the already existed protein-based genome methods. Let us see one by one with the parameters. Mode of action. If you take a mode of action as a parameter, in the case of zinc finger nuclease, it will make a DNA break targeted by protein DNA binding recognition. Here also the same. All the three, it is having a recognition of a DNA breakage, thereby ligation of that breakage, and it repaired now the particular gene getting edited. And second parameter is nuclease design and assembly. If you see here, it is very difficult to make a commercial surface, and it is also expensive one when we are considering a designing and assembly or a synthesizing one. When in the case of talent, it is much more feasible in labor, but it requires more labor intensity and a trained person. Here in CRISPR, it is a high feasible. Easily we can able to design. Nowadays we have various open access softwares to make our own desire guide RNA designing. And the success rate, if we are comparing, the CRISPR is having much more higher success rate over the talent and zinc finger nuclease. And target range, if you take, there must be a limited target range in zinc finger. In talent, it is having unlimited. In in CRISPR, it is having a limited, but it is having a precise. It is precisely able to distinguishing self and non-self, as well as it can able to make a precise cut over zinc and talent. So these are all the overall comparison chart and a table when we are concluding which method is feasible and which method is user friendly, which method is stringency, which method is consistency to our genome editing experiment. So we have to design, we have to consider which method is feasible one for us. Now we'll move on to the methods and tools in CRISPR technology. In before presentation, we have seen the overall basics and natural mechanism of that CRISPR and what are all the elements, components of that CRISPR-Cas9 technology. Now let me move to that practical side. So now we have what are all the methods? We have a doubt in our mind. What are all the methods and tools are available in a CRISPR technology? After designing, how can I do my experimental design? What are all the methods are available for gene transfer, and what are all the points as to be remember when we are designing a guide RNA, when we are choosing a vector expression system. So that makes a sense, and that give you a success rate of your experimental design. Now we'll see the supporting methods and the tools in CRISPR technology. CRISPR-Cas9 system allow many researchers to create diseased model by knockout gene model or knock-in gene model, either in a mice model or in a bacterial cell. So both it is possible. So usually the gene of interest by the use of 20 base pair, guiding RNA and Cas9 endonuclease involvement. So it is easy of use. And another one, it is a revolutionary genome editing system with potentially higher advantage over other protein-based genome editing method. So let me see one by one the gene delivery methods available, and in the gene delivery methods, what are all the category? Viral vectors, non-viral vectors. In gene delivery methods, we have physical, chemical. So those we'll see in our upcoming session. So now we'll see the gene delivery methods. So gene delivery method is nothing but once you are designed your targeted gene, that gene has you have an idea that gene has to be transferred into that living organism to support your genome editing to success your editing experimental design. In that case, you must have to choose your precise delivery vehicle. So based on your experimental design, and here I have given you a parameters like in vitro or in vivo research, size of your gene, target cell type, transient or permeant expression. These are all the 4 important parameters you should have consider while you are choosing a gene delivery methods. While in the case of in vitro and in vivo, you have to choose whether you are going to use a mammalian cell or a prokaryotic cell or a plant cell. As in size of a gene, because not all the vectors can have the carrying capacity, can have the similar carrying capacity. The carrying capacity will be differ according to one vector to another vector. So based on your gene of interest size, you should have to concise about choosing that particular delivery vehicle. Another parameter is the target cell type. whether you are going to target a cell line. It must be a stem cell, embryonic stem cell, or somatic stem cell, or pluripotent stem cell, or a germline stem cell. Based on your cell type, you have to choose your gene delivery method. Another one is a important parameter is transient or a permanent expression. Whether you are going to edit your gene and that particular edited gene should be expressed in consecutively or it should express in a short stretch of time. So that makes a sense, and that place you have to make your selection of that particular gene delivery method. But whatever the method you have chosen, it is having their own advantage and a disadvantage. Hence, it is very important to choose which method is suitable to using as a vector to transfer your gene of interest in a living organism. And keep in your mind, there is no any availability of a ideal vector that can be used for all application. For different application, we have to choose our own and desirable ideal or gene delivery vectors, because some vector will have a carrying capacity of only up to 8 kb, some will have up to 5 kb. So based on our experimental design, we have to go for the gene delivery methods. So let us see one by one. First, we'll move on to the viral vectors. So in gene therapy itself, we have seen the vectors. One is viral vector, another one is the non-viral vector. Viral vector are nothing but we are exploiting the viral cells to transfer our gene. Naturally, viruses having a infection mechanism. So it generally outside the living cells, it must be in an inactive state. That is why viruses are considering as a acellular material. It is living as a an inactivated form in the environmental condition. Once it getting access towards the living organism cells, it get transferred into an inactivated form to an activated form. Once it getting activated, it starts to incorporating its genetic material into the genome of the host. Thereby, without the knowledge, the host genome automatically replicating its genome as well as the viral genome. So thereby it releasing the virus particle, and the resulting is the infection of that viral particle throughout the living organism. So this natural mechanism is exploited by the researchers to use that viruses as a vector. While in this case, we have to edit or we have to alter or we have to remove that virulence gene. Any viruses if you are taking, it must be having a some short stretch of gene. That group of gene or a stretch of gene will have a ability. One must be a virulence, another one will be virulence. A virulence or the nature, they are not infectious to the host system even though they are getting inverted. But this virulent gene will have an infectious nature. Once it getting access to the living organism, it starts to synthesize the cell and it proceed to halt the normal cellular mechanism of that host system. So in the case of gene delivery methods, we have to remove this particular virulent gene from the vector. Now, this viruses doesn't have knowledge, it doesn't have an idea whether it is having the virulent gene or a virulent gene. So, in the place of that virulent gene, we have to insert our gene of interest, which is going to be transferred or replaced in the living system. We have to insert our gene of interest in the region of that virulent gene. Once it getting incorporated, the cloning must be happening in the expression system. That is the clonal expansion of that particular protein will be possible and achieved when they are getting incorporated into the host expression system. The viral vectors, which are all generally used for gene delivery methods, are lentivirus, adenovirus, adeno-associated virus. Each viruses is having their own advantages. And the major one, it is having a infection efficiency. Viruses is having infection efficiency, that is the common feature. is available in the viral vector. Another one, there is a drawback of using a viral vectors over a non-viral vectors. It is having a issue in gene carrying size capability. It is a limited one. Another one, it will create a acute immune response. When we are injecting into a mice model or a human clinical trials, it will automatically create a acute immune response because that is a virus, it is having an antigen. So that once it is getting transported into the living organism, automatically our system will recognize that and considering as a non-self one. Thereby it start a immune response. That is one of the drawbacks available in a viral vector. Another one is the production of a viral vector in a commercial manner is a difficult one. So these three are the major disadvantages we'll faced during viral vector utilization. Let me see lentivirus. Lentivirus can able to infect both proliferating as a quiescent cell. Quiescent cell are nothing but the cells which are all in a G1 phase. That is the silence phase or a senescent phase. Proliferating phase are nothing but the cells are which are all under rapid cell division. So it can able to access to infect both the proliferating as well as the quiescent cell. Another advantage, it is having a long-term stable expression. Once we are inserting our gene of interest and it is getting infected the host system, it is having a capability to express our gene of interest in a long-term manner. This is one of the unique advantage appear in a lentivirus. Another one, it is having a DNA carrying capacity up to 5 kilo base pairs. Until 5 kilo base pairs, we can able to transfer our gene by using a lentivirus. So based on that lentivirus tropism, we can able to re-establish with the different types of envelope protein, which can aid in the production of a viral vectors. So example is vesicular stomatitis virus glycoprotein. So this involved in the lentivirus tropism. So based on the temperature dependent, we can able to modify the envelope protein in response with our commercial viral production. Second, we'll see adenoviral vectors. So this adenoviral vectors can have accommodation of a DNA carrying capacity up to 8 to 9 kilo base pair. Now the entry of adenovirus into the cell is mediated by the highly expressed cell surface Coxsackie virus B adenovirus receptor. In short form, we call it as a CAR. Based on the presence of this receptor only, the entry of adenovirus into the host cell is possible. And it is also having a capability to targeting or accessing both proliferating as well as the quiescent cell. Adenovirus vector is having a transient gene expression and it can only remain as episome inside the cell. It doesn't having a permanent gene expression. It only have transient gene expression. So it is all and it is also having one drawback. It can create acute inflammatory response when we are doing an in vivo research study by gene delivery method, considering this as a gene delivery vehicle. This is one of the limiting factor which is still applicable in a therapeutic clinical applications. Next, we'll see adeno-associated viral vectors. This adeno-associated viral vector lack pathogenicity when compared with lentivirus and adenoviruses, and it is having a carrying capacity up to 3.4 kilo base pair. Again, I'm saying in lentivirus it is 5. In adenovirus, it is 8 to 9. In adeno-associated virus, it is 3.4. So based on our experimental design, we have to choose which viral vector is the ideal one. Now we'll see adeno-associated virus. As I said, it lacks a pathogenicity. It is having a carrying capacity of up to 3.4 kilo base pair, and it also having nature of infecting both proliferating and a quiescent cell. And this mode of entry into the cell is by the presence of heparin sulfate proteoglycans and integrins. These are all nothing but the glycan molecules which are all present in the cell surface. So based on the presence of these only, they can able to bind with that host cell. Once there is a complex formation between the host cell receptor and the receptor and the proteins of this adeno associated virus, thereby it get access, and it the host cell now allow the virus cell to enter into it. So these are all the entry required proteins for the viral vectors. In adeno-associated virus, it is heparin sulfate, proteoglycans, and integrants. In the case of adenoviral, it is a cell surface Coxsackie virus B adenovirus receptor. These are all the three major viral vectors which are widely used in transportation of our gene of interest in CRISPR-Cas9 technology. Now we'll see what are all the non-viral gene delivery methods are available. Generally, it is divided into two categories. One is physical, another one is a chemical. Again, I'm saying each one have their own advantage and a disadvantage. And another one drawbacks in non-viral gene delivery is it is having less efficient in gene delivery, because in the case of viral, they are naturally having that infection efficiency. But this viral non-viral vectors not have that much transferring efficiency when compared with viral vector. Another disadvantage is it doesn't another advantage of non-viral gene delivery method, it doesn't provide any limiting factor for DNA carrying capacity. It doesn't provide restriction on transgenic size and no concern with immunology responses. These two are the advantages over viral gene delivery method. Now we'll see the methods comes under the physical non-viral gene delivery method. One is microinjection, another one is electroporation, and the third one is the gene gun, ballistic. With the help of a gene gun, we have to coat our gene of interest by a conductive material. Naturally, generally it is a cold particles. Once we are coated that cold particles over our DNA genetic material, with the bombardment of that particular gene gun, we can able to forcefully we can able to insert our gene into the living system. It might be either a cell line or a plant cell. Either case it is possible. And another one, it is a micro injection. In micro injection, we'll use a needle. Inside the needle, we have to take our gene of interest. So in other hand, we have to hold the cell with the help of a holding catheter. With that help, we have to insert our gene of interest. With the help of this injection, forcefully we can able to insert our gene into that cell by using this method, micro injection. Another one is the electroporation, which means with the help of electric field, around some 100 to 250 volt, we can able to give the electric field. Once the environment is created by electric field, the bacterial cell will make a pore in that cell membrane in response with dynamics, that is the molecular dynamics of the cell membrane mechanism. They are creating a pores in the cell membrane. We are utilizing this condition. At this condition, immediately we have to transfer our gene by making a competent cell preparation. Competent cell preparation followed by electroporation will give a high successful rate of gene transfer capability when we are considering non-viral gene delivery method. So these are all the commonly three available methods for gene transfer. Now we'll see chemical non-viral gene delivery method. Here we have two methods. Calcium chloride, another one is a liposo. So in calcium chloride, as I said, that is the competent cell preparation. We have to give a shock. We have to give a temperature shock by altering the temperature. That is cold temperature. Initially will give a incubation temperature of 37 degrees Celsius. After the transferring, after incubation, we'll give a calcium chloride treatment, followed by there is a heat stack at some cold chilled condition, around some minus 20 degrees Celsius. We have to give a cold shock. At that time, the cell will form a condition. That condition we call it as a competent. That it is having ability to accept any of that foreign DNA which is not belong to that particular bacterial DNA. It is forcefully placed in the place where it is able to accept all the foreign genetic material. Followed by the heat star, will have our gene of interest. Automatically, that particular bacterial cell will accept our gene interest, and thereby followed by there is a another incubation time period to allow that cell to set up. Once there is a successful transfer of our gene, we can either go for a blue-white screening colony techniques to check whether our gene is completely transferred or not. So, followed by competent cell preparation, we can go for screening and validation technique of that particular blue-white colony technique. We can able to validate whether it is getting transferred or not. This is one case. Another case is the liposome. Liposome is nothing but is like a measles. The outer cell will be a hydrophobic in nature, inner will be a hydrophilic in nature. Generally, our DNA, that is the nucleic acid, is having negative charge, which means it is having a phosphate backbone, hence it is having a negative charge. So it is masking this liposome, masking the charge of the DNA, and it also provides compressing of that stretch of a DNA into a concise one. Another one function of this chemical method, it protects the DNA from degradation. Naturally, in all cell, extracellular nucleases is abundant one. Whenever it finds a nucleic acid, it cleaves that. If our gene of interest is getting cleaved, there is no any validation to our experimental works. So to avoid that, we can use a liposomes. So these are all the three functions, and if you see here, it is having, due to hydrophilic nature of DNA, it cannot efficiently enter into the cell naturally. So that this liposomes will aid the way, because as I said, it is having an outer hydrophobic region, inner hydrophilic region. So thereby it is having easily get access into the cell, and thereby the gene transfer is getting access. And another one, it also involved in electrostatic interaction, that is positive and negative charge interaction. And there is one disadvantages in chemical non-viral gene delivery method. It is having a short-term gene expression. It is not like in the case of viral vector can have the ability to permanent expression or long-term expression. But in the case of non-viral gene delivery, it is having a drawback of short-term gene expression. Only a short time of period only it can able to express our gene of interest, not a long-term manner. So these are all the available gene delivery methods when we are doing an experimental design. Now we'll see the designing of guide RNA. Up to that, we have seen the basics of CRISPR technology, and the gene delivery methods are available for transfer of a gene. Now we are moving to the third session of our webinar, that is the designing of a RNA, guide RNA. We'll see one by one. So this guide RNA, as I said, it is a complex of CRISPR RNA, tracer RNA. So The most commonly used guide RNA is having a short stretch of 100 nucleotide base pair in it. In that 100 nucleotide base pair, there must be a 20 base pair is complementarity to that template strand. That template strand is nothing but the strand which is incorporated from the invaded foreign particle. So there is a complementarity, that is the 20 base pair complementarity between the targeted template strand and a guide RNA. The CRISPR-Cas9 system can be used to target any genomic region which is complementarity to that particular sequence. This is the one of the most important point while we have to consider doing a guide RNA designing. We have to choose the complementarity sequences of the targeted gene which has to cleave, not a non-targeted region, because this guide RNA as the nature, wherever it find its complementarity region, it go and bind that and it recruit a Cas9 nuclease and it make a cleavage. So we have to very much concise and precise while choosing and designing a guide RNA. Let us see what are all the guidelines which are all available in a guide RNA design. So targeting a specific region in a genome which can be only achieved with the help of that complementarity sequence, that is the 20 nucleotide sequence which is present in the 5' end of the guide RNA. Now this guide RNA guides Cas9 nucleases to the target sequences by means of complementarity base pairing. Once there is a bind complex of guide RNA to the template strand, it recruits Cas9 nucleases and thereby it mediates the double strand breakage at the 3' nucleotide upstream of the PAM sequences. Why? This guide RNA will recognize this PAM sequences and thereby it bind to that particular template strand by means of complementarity base pairing. Once there is a complex of PAM, guide RNA, template, these three complex will recruit Cas9 nuclease and thereby aid the double strand breakage at the DNA of viral particles or transposable or bacteriophages. Just remind, PAM sequences is not belongs to the part of a 20 base pair of a guide RNA. Actually, it is a short stretch. This PAM sequences must be having a length of around 2 to 6 base pairs generally. So, in some case, promoters are there. It may this may not always telling gene expression. It may also lead to repression activity, because if there is a particular concise promoter is not present, the activity of that gene expression of tracer RNA, transactivating RNA and CRISPR RNA getting inhibited. To avoid that, we have to choose ideal promoter. For example, if we are using a plasmid for transporting, we have to choose a ideal promoter which can aid efficiency, high efficiency rate in the gene expression and a gene regulation pattern. To make a sense, we have to choose our ideal promoter. In case, for example, I'll give you an example. This is a U6 promoter. This U6 promoter is a RNA dependent, RNA polymerase 3 dependent promoter. For example, it is present at the 5' end of the RNA sequences to initiate the transcription mechanism. In such case, this promoter commonly used for driving a small hairpin RNA expression. Example in a RNA interference vector-based method. While designing and construction of a viral vector, we have to consider transcription unit arrangement in close proximity in a space-limited manner. And another one, this U6 promoter normally control expression of U6 RNA, that is the small nuclear RNA, which is involved in splicing and splicing mechanism. And this particular promoter can be considered and chosen as a control vector based expression of small nuclear molecules in mammalian cell. Usually, we know promoter or the controller session of the controller point of the gene expression. When there is an inhibition of the promoter region, there won't be a possible of gene expression. So, in a gene transcription unit, if you take, there is an arrangement of promoter region, followed by there is a effector, there is a inducer, and there is a structural region, there is a terminator. Once there is a complex formation between a promoter and a DNA polymerase, the gene expression will get on. If there is a repressor is bind with that promoter, there won't be a gene expression. Automatically, it turned off the gene expression. So that basics is only applicable while we are designing a guide RNA. So we have to choose our precise promoter while choosing a vector. This is one of the important factor in guidelines of CRISPR-Cas9 technology. Another one, while we are doing a designing in software, we have to give that particular synthesized and designed software to synthesizing company to get our synthesized guide RNA. We have to make sure about these 3 points. First one, GC content. GC content, that is the guanine cytosine content, it should be in a range of 40% to 80%. In case if there is a high GC content, it can stabilizes the RNA DNA duplex rather than destabilizing the off-target hybridization. If in simple way, I can explain. When there is a higher GC content in your designed guide RNA, it may create a high strong affinity, but it may create a more error, and it also develop more off-target cleavage. So in that case, we have to make a point. Our guide RNA should be in a concentration of content of 40% to 80% of our GC. And another parameter is a length. So that guide RNA should not be beyond 100 nucleotide base pair. Generally, it should be 70 to 24 base pair in a length is the ideal one. And it also screened for off-target sites. So there are n number of softwares are available to categorize our synthesized RNA or a designed RNA, whether it is having a high number of off-target cleavage sequences or a low number of off-target cleavage sequences. Examples like I have listed out here: Chop Howard, CRISPR Design, UPA GDT. These are all the available softwares, and all the available softwares, and this Chop Howard and UPA GDT are all open access. So there is no need to pay to design your guide RNA. You can use and you can access your optimized one by using this software. This Chop Howard software analyzes the sequences and identify almost all possible 20 base pair sequences, which are all immediately present, followed by the PAM sequences. It also scores your guide RNA according to your preset score, which is programmed in that software, which looks for all these 3 parameters. It looks for GC content, and it looks for ideal length, and it also give you a data of potential off-target sites in your desired gene. So based on the score given by the software, you can choose your optimized one. So that particular sequence is copied, and that copied you can send it to your synthesizing company to design your guide RNA. And also there must be some disadvantages also is that if you are using a CRISPR design, it can able to screen base pairs of about 500 base pairs at a time. So this is a disadvantage we face in a CRISPR design, but it is recommended to use a chop forward rather than use a CRISPR design. So now we'll see fourth session of our webinar, that is the screening and validation. Even though we have successful gene transfer, we have successfully designed our guiding RNA, the experimental design will always depend upon the validation side, the study validation side. This study validation site is nothing but it is having some parameters or a checking checkpoints. So that we can able to check whether our all methodology and tools are operated in a correct way or error-free manner. So although CRISPR system for genome editing is efficient, it is not ideal, and it can't able to do the edit in all the population of the cell. As we know, if we are doing a edit in bacterial cell, it is very highly efficient one, it giving high success rate. But in the case if you are using eukaryotic cells like animal model or a plant cell or a human cell line or a stem cell, in that case we have to choose, because if a live animal model if you are choosing for our genome editing is a mice example, I'm telling mice, it is having a it is having many number of cells. So we can't able to predict our particular genome editing will be acquired, will be possible in all the population of the cell. So we have to precise about our experimental design, and we have to choose and we have to screen and validate our experimental design. So for that we have to choose four main categories. One is on target mutation. So after gene transfer we have to screen whether there is a mutation happening in the genome. Second, there is a screening method. There are n number of methodologies are available: Sanger sequencing, NGS, droplet digital PCR. So we can either one choose, and we can screen for the deletion and insertion mutations. Another one, screening of cells edited with a homology directed prepared template. Last one is the screening of the indels. These four are the parameters which are all considered while we are screening and validating our CRISPR-Cas9 study. Here I have listed out some indel screening methods are available in CRISPR. One is mismatch cleavage detection method, and where we are utilizing surveyor nuclease assay. But thereby we can analyzing the mismatch or mismatches of single base pair or small insertion or deletions in the genome. Another method is high resolution melting analysis, where this method is relatively new one, and also it is happened after post PCR analysis method, and this method can use to identify variations in the nucleic acid sequences by reference with the PCR melting dissociation curve. And another method, if we are seeing CAPS, CAPS is nothing but cleaved amplified polymorphic sequences, nothing but the designed single guide RNA, so that if there is an indel, that is insertion and deletion, is formed, it will disturb an enzyme restriction site and then use restrictions enzyme digestion for screening. So this method will give you a enzyme disruption site. So thereby we can able to identify whether there is a polymorphic sequences happening after our insertion of our gene of interest. And another example is loss of primer binding site. For example, if you are inserting our gene, and in such case, it might create some overlaps in a putative insertion or deletion site, where it may disrupt the function of a particular gene, and it may lead to the attachment or binding of our primer. So that in that case, we can screen our guide RNA. It is that is screen our guide RNA in response with that losing of primer binding site. Another one is a droplet digital method. This utilizes water droplet technology method. And followed by we have a fluorescent PCR capillary gel electrophoresis. This is nothing but coupling of coupled use of fluoropore primers. That is fluoropores like fluorescent tags or attached with that primers to generate an amplicon. Whenever there is a formation of a PCR product, it will emit a light. Thereby, the detection will detect that fluorescence signals, and followed by it is coupled with resolving the PCR products by capillary gel electrophoresis. In such case, we can able to identify indel mutation shows in a different mobility to wild type and designed synthetic one. Just hold for 5 minutes. I'll be back in just 5 minutes. முடிஞ்சிருச்சு ma'am. Sorry for the inconvenience. Sorry for the inconvenience. Let me start the presentation. So, in previous slide, we have seen some parameters and screening and validation techniques, which are all we can use when we are doing a CRISPR Cas9 experimental designs. Now we'll see CRISPR Cas9 technology case study. So it is like one of case study which is already done by using this CRISPR Cas9. We can is a bacterial gene knockout case study. We'll see what are all the phases we have to follow by doing a knockout gene methodology in a E. coli cells. Actually, this is followed by the knockout of a gene that will code for a chloramphenicol resistant cassette. Actually, this gene is getting knocked out from the E. coli cells. Now, this E. coli cells is grow on a chloroform added Petri plate. At that case, it will not grow on that particular growth medium. Rather than it will grow on a medium that lacks a chloramphenicol antibiotics. So this is one of the case studies done by the researchers by using CRISPR-Cas9 methodology. First step they have did designing of a Cas9 enzyme, single guide RNA, and the expression of this Cas9 and single guided RNA into a cloning vector. That must be a expression vector, either it is a bacterial cell or a in vivo cell lines. Second phase, they done preparation of a red induced competent cells to accept the designed single guided RNA Cas9 and our gene of interest. And once there must be an insertion of our designed gene, and they are performing a transformation by followed by the competent cell preparation. Third one, they are validating their experimental design by testing that by growing that particular bacteria on the chloramphenicol antibiotic added plate. So in that plate, what they did, they have added both the knockout chloramphenicol resistant cassette E. coli cells, as well as not altered or not removed chloramphenicol resistant cassette E. coli cells. Two cases. One E. coli cells is a knockout of chloramphenicol resistant cassette. Another E. coli cells is doesn't undergo any genome editing. It have that cat that is chloramphenicol resistant cassette gene inside that genome. Now researchers have grown these two cells on a two different plates. One plate with a chloramphenicol antibiotic added plate. Another one with a another plate which lacks a chloramphenicol antibiotic. So they were grown and they can able to visibly see the cells which are all lacks chloramphenicol resistant gene can't able to grow on that particular growth medium which have supplement of chloramphenicol. In the other case, the cells can able to grow on the medium which have chloramphenicol antibiotic resistant gene. So this is one of the case study which is done by the researcher by using CRISPR assisted knockout methodology. So the same method is followed for a mammalian cell. In such case, there is a differences. One thing is the delivery method and Cas9 and single guide RNA design. These two will only get differ based on our experimental design and what type of vector we are choosing, what type of hosting that is a host expression system we are choosing. In such cases, we can able to differentiate the utilization of Cas9 and a single guide RNA designing and cloning. Now we'll have some basic idea and not basic idea. I have given some in-depth knowledge about that guide RNA designing in previous slide. Now, if you are doing a designing or if you are frame out a genome editing flow chart, they have some advantages. It should be easy of design. There must be an experimental workflow will be in an easy manner, and there should be a efficiency and off-target effect, flexibility, and a application. So based on these two parameters, we have to concise our points which method we have to choose, whether it must be a meganuclease or zinc finger nuclease or TALEN or a CRISPR. So based on our experimental design, we have to choose. But till over all these three methods, meganuclease, zinc, TALEN, this CRISPR Cas9 is having a high flexibility, more wide clinical and therapeutic applications, higher efficiency in genome editing, and the consistency and easy of design is very precise and a customizable one. So this is one of the advantages of CRISPR Cas9 over all other genome editing tools. So if you are doing experimental planning, if you are going to edit genome to create some transgenic crops, in such case, I'm giving it's like an example. So at that time, we have to think, we have to design, we have to write our own flowchart. First, what you want to transfer and its nature, whether we have to transfer a large size of a DNA, or we have to transfer a short stretch of DNA or a repeated units, what we have to transfer and what its nature. In nature, it's mean by the size and its GC content and its transferring efficiency and its chemical modifications. Those things we have to think about it. First point. Secondly, we design single guide RNA for your target gene. Whether a particular single guide RNA is already available in a commercial one. If not, how we have to design our single guide RNA, and what are all the points we have to make a consideration while preparing a or while designing a single guide RNA. So that makes a sense here. Third point, delivery of single guide RNA and Cas9 into a experimental model. Delivery of our single guide RNA and Cas9 nuclease enzyme into our experimental model. This experimental model is nothing but the cell type, whether it is a cell line or a stem cell or a in vivo model like a mice model or a crop. So that we have to choose ideal gene delivery method. And fourth, we have to evolution of its efficiency by screening and validating our experimental and design guide RNA, or overall our experimental design we have to screening as well as validation, because this validation will only lead to the high success rate of our experimental design. These are all the points we have to keep in our mind while we are designing a CRISPR Cas9 experimental works. Now we'll see the end of our webinar, the ethical and social and legal issues which we are all facing in a CRISPR technology. Even though it is having a wide range of applications in clinical as well as therapeutic applications and commercial side and research and development side, it is having some ethical and social issues while we are utilizing that. CRISPR could be opted for many purposes, like bioterrorism also it plays a role, biowarfare. So it's nothing but it is in our hands for what purpose we are using that genome editing. That makes a sense. It doesn't naturally having a capability to become a bioterrorism or a biowarfare. Actually, it's based on our human handling. So based on that, that thing is also one of the ethical issues while we are considering CRISPR-Cas9 as a genome editing tool. Another one is it can engineer vaccine-resistant flu viruses or invasive species, and this can also, while doing a research works by utilizing this CRISPR-Cas9, there is an opportunity to create a non-pathogenic microorganisms into a pathogenic one. So we are creating or we are giving a opportunity to that particular microorganisms to convert it into a pathogenic one. So that is also one of the ethical issues while handling CRISPR technology in a crude laboratory. And third important point in that here, who and how informed consent is obtained. Informed consents are nothing but the human clinical trials who are all accepted to involved in trialing of that CRISPR-Cas9 technology for therapeutic applications. We have to make a convincement and we have to explain our research study to those informed consent, and we have to choose the informed consent based on the health category as well as the study experimental design category. So we have to choose whom and how informed consent is obtained. Then another one, in India, there is a restriction to do a genome editing in embryos. We can do our genome editing in somatic cell lines, but we are not allowed to do our genome editing in embryos. The major one, which is restricting not to use germline is germline cells are having the ability to transfer from one generation to another generation. For example, if there is any mutation in accidental case while we are doing a CRISPR editing, in accidental case, if there is any insertion or deletion in a non-targeted region, and that non-targeted region will aid a role in important cellular function. At such cases, that particular germline will have the ability to transfer that error to that next generation. So the germline embryos and germline cells are not naturally allowed to edit by using a genome editing tools. In somatic cells, we can do our editing, but still in India, it is not advisable and not allowed to do our editing in a human embryos. We can do editing in bacteria and mice model and plants is possible, and even in animals also they are acceptable now. And in another one ethical issues faced by CRISPR, it can create a large DNA deletion without a knowledge. If we are not, if that particular insert and deletions are not identified in, even though in screening and validation, that may also lead to large DNA deletion that created a undesirable phenotypic changes and genotypic changes in the whole genome. The overall success rate of CRISPR technology in a therapeutic application still it must be in a range of 1%. Might be increased if we are using much more identification of a novel CRISPR-Cas9 system by identifying a novel bacteria and based on synthesizing and optimizing of our guide RNA, we can able to increase this success rate. If you see here the bioethical issues of CRISPR-Cas9 technology in the category of bacteria, it may create not only in bacteria, in all organism, it creates non-target mutation and leads to genetic drift. There is a population disturbance between one gene flow to another gene flow generation. So that makes a considerations of bioethical issues in CRISPR-Cas9 technology. Another one, it may create an ecology imbalance. For example, naturally in a microbial environment, there must be a set of microbes out there. If you are doing a CRISPR-Cas9 in one category or one cell, it may lead to division and it may create some new population. In such case, it will create ecological imbalances, and it is opposing to that natural one. So that is also bioethically not accepted when we are considering CRISPR-Cas9 technology. And in humans, it is very difficult to make informed consents, and acceptability of patenting is not available if we are doing our genome in human and animals. In bacteria, we can file a patent, but while we are doing editing in animals and humans, it is not allowed to patenting. And another one, there must be a safety issues is also that if we done any genome editing in a important gene regions. And also it might face some law and legislations if we cross the rules like genome editing is not involved in germline embryo. If we do such editing in embryo, it must be leads to legal issues. So these are all overall the ethical and social issues we faced in CRISPR technology. Even though CRISPR Cas9 is a tricky concept to explain, but especially concerning the potential of target genome editing, this CRISPR Cas9 technology is sprinkle and shining like a one of the wonderful evolutionary tool over and masking all these disadvantages. So it giving a addition of customized one. We can able to creating a customized gene editing on the top notch that will further push the experimental design into a higher success rate. So that makes a sense to use CRISPR Cas9 technology as a genome editing tool. So these are all the therapeutic applications which is currently applied with the help assisted of CRISPR Cas9. In cancer therapeutics, we are using CRISPR Cas9 technology to treating cancer cells like lung cancer or blood cells, so that to eliminate resistance of that particular cancer cell, because cancer cells have some resisting gene that may inhibit and resist the chemotherapy. In such case, the CRISPR Cas9 system is used to inhibit that resistant gene to increase the efficiency and responding of that human body towards the chemotherapy. That is one of the application of the CRISPR Cas9 in cancer treatment. Another one is agriculture. In agriculture, we can use this system to create crops which can provide high yield varieties with superior quality, and they can also have some herbicide resistant, pesticide resistant, or a salt intolerance. So these are all the characters generally farmers required while doing a crop practices, because they always should aware not to decrease the yield of their crops. So in such case, by using this CRISPR Cas9 technology, we can enhances the crop production. Other application, if you see here, this CRISPR Cas9 technology can also be acting as a regression model. We can using this as a live imaging. For example, we can fuse the fluorescent tagged protein like GFP, green fluorescent protein. We can tag this GFP with guide RNA. So wherever this guide RNA go and bind the template, this fluorophore will get emitted, and this GFP will get emitted. There must be a reporter gene will give you a resulting live imaging of that particular DNA and mRNA binding proteins, or functionality of that DNA we can easily able to trace with the help of this CRISPR enzyme. Another one, we can also do epigenetic editing and screening of DNA libraries and other genome editing to create knockout and knock-in models, transgenic crops, transgenic mites. These are all the other applications of the CRISPR technology. I hope everyone will have some basic outline of a CRISPR technology from basics to advanced level. In this webinar, we have framed out. So this webinar is covered overall the basics, the nature of a CRISPR Cas9 technology, and how it is used and how it is utilized, and how the guide RNA should be designed, and everything is concised in this webinar. I hope everyone will enjoy this webinar, and everyone will have some idea about CRISPR Cas9 technology. So now let me conclude this webinar by exploring our upcoming events. If you want, you can also join in our upcoming events. One day workshop on role of microbes in agriculture, In this workshop, we will tell what are all the important microbes in which is taking place in agriculture, and we have a 7 days internship on real-time PCR in a practical session. Actually, we'll give both theory as well as the practical one. We'll cover both the category, and in practical session, we'll give how to design a real-time PCR mixture, how to set up a reaction, how to interpret the reaction. Everything will be taught teach in that internship. And another internship, if you see, that is on molecular visualization. In molecular visualization, um, we have used wide number of softwares to visualize the docked protein and a ligand, and also we will cover what are all the favored contacts between DNA and a protein, and what are all the unfavored contacts between protein and docked protein and a ligand. Those things will be covered in this internship. And every month, not only this November, in every month we have a release of our own e-magazine. In this, we are encouraging young talented minds and upcoming generations like students. We are encouraging to submit their research articles, short articles, communicating articles, or if they have any idea about particular topics, they can also write some write-ups about that particular topics. They can give us an article. We will publish that in our e-magazine, and followed by we have a ISBN indexed publication. This is a quarterly, half quarterly publication book release. So we are welcome all authors to submit their research articles, review articles. So this will give you a CV weightage, because we are giving a ISBN indexation to each author, to our book release. So each author can use that particular ISBN indexation to index the review or research article. And other than that, also, we are also providing a 3 months discussion project like drug discovery and plant therapeutics. And we are also given 3 months project on secondary metabolite production, on followed by GCMS interpretation data. So those things are also um, done in 3 months discussion project. But in upcoming, we are planned to release a 3 months discussion project on drug discovery. So you will have a hands-on practical session, as well as we will give a guidance to publish the paper in a high impact journal. So mark your interested topics in your calendar, and do your active participation in those upcoming events. So at last, on behalf of Veridian Microlab Private Limited, I hereby completed my webinar. I hope everyone and every participants and every delegates and the visitors who are all watching this webinar will have in-depth idea about that CRISPR-Cas9 technology. If you want to learn much more in a practical session, we are also providing an internship in CRISPR-Cas9 technology. 7 days internship program is also there, so you can also access through it. If you have any queries, please write your queries to email info@veridianmicrolab.com. We are always available to service you. Thank you. And participants can ask their questions if there is any doubts. You can write your questions in a live chat. We will see and give you a responsible reply. I have a question. Subrata Ghosh. In genome editing, both guide RNA and case proteins are engineered and programmed. Now, how it is related to E. coli or Clostridia species? Actually, I have framed out. You can choose both the model system E. coli or Clostridia. This guide RNA or case proteins are isolated from a bacteria only. So in such case, if you want to do your genome editing, you can choose your case 9 protein category. There is three categories are available in case nucleases. So based on your requirement, you can choose. As I said before, this case 9 nuclei and this CRISPR array will not be same and will not be expressed similarly in all the bacterial species. So E. coli will have some different guide RNA sequences and case 9 proteins. Clostridia will have some different sequences of for guide RNA. I'm mentioned that model organism to mentioning that guide RNA and a case protein, um, not for a programming cell. And Jaidev Sharma recently attempt to use CRISPR technology to modify embryos and make them resistant to HIV. Given these disadvantages, that doesn't make sense. Actually, there is a even though they have tried CRISPR technology in human models, still now it is not considering in a therapeutic application. Still, it is in a research and development platform only. Not it beyond that. We can like without any doubt we can able to use CRISPR Cas9 in a therapeutic application of HIV treatment. Actually, that makes a sense there. And still there are many papers and there are many um, studies or are outlined by assisting this CRISPR technology. Recently, one of the paper is also done in human embryo, but initially they were accepted in publication in one high impact journal, but later after revising they were rejected that paper. Still, it is available in research and development part only. And Subratto Ghosh, repair method is something like recombinant DNA technology. Of course, but not exactly with that. Actually, repairing mechanism is when there is a breakage in double strand or a single strand, the host cell it has to repair it before it the cells undergo a cell division to prevent a mutation, to prevent a unwanted genomic expression. The cell has to repair that particular DNA. So HDR is a much more concise over non homologous end joining method. Mm. Any other questions? You can write it in a live chat. Any questions? Okay. So I hope as much I can provide the information about CRISPR Cas9 technology. If there is any more doubts, you can write your doubts to info@veridianmicrolab.com. And now I hereby close our today's webinar. And there is a one notification. This webinar is having a e-certificate, so which is based on your quiz score. Actually, we have shared our link in a live chat, and this link will be activated until up to today 11:59 p.m. After that, the participants who are all using this link and those who are attending the quiz will not receive the e-certificates. By today itself, up to 11:59 p.m. is the acceptable time to submit your responses, and thereby, if you score or cross the passing score of 40%, then you will receive your certificate. Those quiz questions are completely based on our today's webinar. Okay. So we Veridian Microlab Private Limited, we hereby closing this warm evening with the completion of that webinar. And if you have any doubts, please write it to us. Thank you.